everyone, my name is Professor Brittany Gale, and I will be doing your videos for this semester. Hopefully these will help you as you study and that we can both have a successful journey this year. Anyway, let's get started. So we'll begin with Chapter 16, the evolutionary history of life on Earth. So, just throughout the video I'll move myself. Anyway, science and social responsibility. This is something I like to start off anytime we're talking about any sciences. This is for chemistry, physics, anything that's dealing with science and the scientific method. Science is awesome. It helps, has it helped us with our lives? We have vaccinations, we have genetic research. We're able to learn so much. We have all this technology. The death rate has fallen, all these wonderful things. But I do want to point out that science is impartial, and it simply attempts to study the natural world. It does not make ethical or any moral decision. Science cannot prove anything. It can show that things are falsifiable, meaning that science can't prove something to be true, but it can prove something to be false, I suppose. So that's kind of what we need to go off. It does not make any ethical issues. Should we do something? Should we not? Is this a truth? No, we can't go there. So that's something I just want to point out anytime I start any classes, that we have that science and social responsibility. You have the knowledge. What you do with it, that is something that is that moral ethic barrier that we need to make sure we always stay on and have a good responsibility. Anyway, so chapter 16, here's the outline. We're going to talk about evolutionary history of life on Earth. And so there's a couple characteristics that make what something is life. Then we're going to get into the Linnaean cladistics and different taxons. We're going to talk about 3 different domains. Then we're going to get into systematics and cladistics. And so that's kind of what we're going to be going through today. All right, so what makes something alive? Why is that we say alive when that's not? Well, there's a couple of things. First, everything that's alive is made up of cells, and those cells are organized. So all life forms have a cellular level of organization, and these can be either single-celled or multiple cells. So things can survive as being one cell, and they're still alive. Okay, everything that's alive has to metabolize and undergo what's called homeostasis. So this means you have to intake food, and it has to convert into something that can be used as energy. It has to be able to form something that it can use to work. It also has to be able to grow and repair itself and reproduce. Homeostasis is the balance between the outside environment and the inside environment, making sure everything is fine. You don't have too much, let's say, salt in the system on the inside versus on the outside in the environment. So that's kind of what that means. 3, everything that's alive responds to stimuli. It reacts to the environment for the purpose of survival. So when you touch something hot, you have a reaction. When you hear a sound, you react. When somebody bumps into you, you do something. Those are what the stimuli are talking about. So anything that can react to sunlight or anything, that is an indication that it might be alive. 4, it must reproduce. Now, sexual or asexual selection, and we'll talk about those in a minute, but sexual or asexual reproduction is for the purpose to perpetuate of the species. So it has to make more of itself. And then the last one, 5, it has to be able to evolve and adapt to the environment. So this leads to diversity, and this is strictly for the purpose of survival. So you evolve and adapt as a population in order to keep thriving. So those are the 5 things that are necessary to make something alive. So let's talk now about this gentleman. His name is Carolus Linnaeus. He came up with a way to classify and name organisms. So we call that system taxonomy. And this is the part of biology that is talking about when we ID something and we name something and classify it. And so the universal language is Latin. Why do you think that is? We'll talk about that in a minute. Anyway, so Carlos Linnaeus, he was accredited with what's called the binomial system of naming. And this means bi, meaning two. So this is a two-name system. And so every living organism that has been classified will be given two names. And we'll talk about how that is formed. And so species are going to be classified in categories, or what we call taxons, based on their physical and biological similarities. Okay, so let's talk about this binomial naming system. So you have the scientific name. So you have two parts. You have the species name, and that will become developed by having what's called the genus and the specific epithet. Now, you have heard this before. If you've heard of Homo sapiens, that's exactly what it is. The genus is one of the taxons that we'll go into detail more, but you have the genus first, so that is Homo. And then you have the specific epithet, sapiens. And now, sapiens is not a word on its own. It's just a description, and it doesn't mean anything by itself. So you can't just say, oh, there's a sapien. You always have to have both. You have to have Homo sapiens in order to talk about humans. So there's a couple things to note. Homo sapiens, you have to have both to make a name. Second, it has to be italicized or underlined. That is the proper way to write it. So anytime you see any name of an animal or a plant or anything that's alive, has to be italicized and or underlined. Also, the genus is always going to be capitalized. And so the whole purpose of taxonomy is to provide a common language for scientists. So this is why we use Latin and Greek, so that everybody, if you're talking about Homo sapiens in the United States or you're in China and you say Homo sapiens, everybody knows what you're talking about. So let's talk about now the different taxons, so where that genus kind of comes from. So we have 8 categories, or what we call taxons, for naming. I'm going to teach you with the most broad, so this is inclusive. There's only 3 groups. So anything that's classified as being alive, it's going to be in one of 3 domains. Then as you go down the list, it gets more and more inclusive. So you start to eliminate different things get more specific. And so the very first one is domain. Then we have kingdoms, phylum, class, order, family, genus, species. And so this is when you get down to the species with the 2 naming system, the binomial nomenclature. With the species, you have to have the genus plus that specific epithet. So if you're in the same species, you're closely related. If you're in the same domain, you're a little bit related, but you're still related. So for this class, you're going to need to know the order. And so I have a little saying how I remember the order. So it's do kings play chess on fiberglass stools. So that's how I remember. So when I'm in my head thinking, okay, what's next in the list? Do kings play chess on fiberglass stools? So do's, domain, kings, kingdom, so forth. So that's how I remember the order of the different categories or taxons. All right, so you guys have probably seen a black bear or heard of a black bear before. And so let's go and look at this kind of chart real quick. So if you see over here where my mouse is, it's domain. So it's the most broad. It includes tons of things. So you see here, you see the black bears, all different types of bears. You have the cheetah, wolf, dolphins, all the way down to butterflies and birds and fungus, and even a little paramecium there. And so that is domain. So it's very, very broad. Now this domain name is called Euchera. All right. So then we're going to go down the list and get a little bit more specific. So then we have the kingdom. As you see, this is kingdom Animalia. So we got rid of all the plants and the parameciums, which is a protist. So we'll get rid of those. Now let's get more specific. The phylum is Chordata. Okay, so this is anything that's what we call a chordate. So it has some special traits that we'll learn later in the semester. But it has, essentially, it's the backbone and a couple of cool things like that. Then we get more specific, the class, which is Mammalia. Okay, so you have mammary glands. Then you get down to order, which is carnivora, so those that are carnivores. Then family, it's Ursidae. Genus, Ursus. and then we get to the species. So to get to the American black bear, it would be Ursus americanus. You see how it's lowercase for the americanus is the specific epithet, but Ursus is capitalized, and that's from the genus. So that's how you get very specific in finding your specific organism. You can also look at it this way with the cheetah. Or, sorry, this is a leopard. And so this is domain, kingdom, phylum, class, order, family, genus, species. And as you see, they have special names that go with them. It's the same, it's very similar to the black bear here, all the way until we get to the, right here, the order is the same, and then we get to the different families, how this is Felidae. Over here, you can say this is Ursidae. So that's when it gets a little bit different when we talk about in this different, this group with the cats. And so you can start seeing hopefully the differences and how it starts broad and it gets all the way down to being very specific. Here's another one with a couple different animals, so you can kind of see what's happening here. So we have the same, we get down to the order, and then as we saw earlier, the family, which is Ursidae, Felidae, and then this is the Canidae, are different here. And then as you see, they get more specific until you get your specific species. So that's kind of how this taxonomy works, the binomial nomenclature. Okay, so now we're going to talk a little bit about the different types of here that are going on. So we have something called a character. Now a character is a biological term. It means any structure, molecular, reproductive, behavior trait that distinguishes one group from another. So what makes this different from this? We call it a character. And so just so you know, we actually have more than 30 different taxons used for different classifications. We have a lot of what's called like the super and sub and infra order, besides just the basics of, oh, this is an order and family and whatnot. So there is a lot more that goes into it, but we're not going to go into detail. I just want you guys to know the basic 8 taxons for this class. So we're coming back to this kind of question again. When we research publications, anytime you look online, even Wikipedia, okay, you can see on the right side of the screen, it goes through the different taxons. And we always refer to an organism by its scientific name versus the common name, like black bear. Why do you think that is? Well, the major reason is to reduce confusion. Have you guys ever heard of a firefly or a lightning bug? What's the difference? What about a locust or cicada or grasshopper? They kind of look the same, and somebody like a roly-poly or a potato bug, they might be the same name or different. It gets confusing. And so the whole reason why in science we use the scientific name is to make sure we know exactly what we're talking about. Also, I wanted to double go over, I guess, with Latin and Greek. These are dead languages. Greek not so much, but Latin, it's not going to be changing anymore. And so if we always stick to the Latin-based language, we're never going to have a problem 20 years from now when a new word is invented, because Latin's dead. It's not going to be changing. So it's always going to be the same, and so we don't have to worry about issues with naming things. All right. So now we've talked about different taxons and everything. Let's get a little bit more organized and learn what these mean. So there are 3 domains. Okay, I think for the years that I've taught this class, this is something you just gotta memorize, and people always get confused. So you have 3 domains, and then we have 4 kingdoms. So domain, kingdoms. Remember, do kings play chess? Okay, so the most broad is a domain. And the 3 domains are bacteria, archaea, and eukarya. Okay, so those are the 3 you need to know. Now, all the kingdoms that you need to know are specifically going to be under eukarya. So when we talk about bacteria and archaea, that's it. We're just going to talk about the domains. We're not going to get any more specific than that. But with eukarya, that's when we're going to get all the way down to this species. So make sure you know there's 3 domains. And under domain eukarya, you have 4 kingdoms. Okay? Now those kingdoms are Protista, fungi, plantae, and animalia. So this box down here is just showing you some a picture review of it, so because I'm a more of a visual person myself. So right here you have the 3 domains of life: bacteria, archaea, eukarya. Now the 4 kingdoms you must know are under eukarya: protista, fungi, plantae, animalia. All right, so here is another view, and this is essentially the whole Bio 124 right here on this page. And so you have up here at the top, you have the domains, you go down to kingdoms. Then we're going to talk about the different cell types, the complexity of organization, the types of nutrition, how they move, life cycles. This is all what we're going to be getting into this semester. So the domains specifically, the 3, bacteria and archaea, then you have eukarya. So down here in the kingdom, like I said, we're not really going to be going into kingdoms or anything specific with these 2 domains, but it is going to be chapter 17. Okay. Now we're going to learn about their cell type, which is prokaryotic. We'll go into details about all this here in a minute. It is also, they are unicellular, so their whole existence is one cell. How they eat, they are autotrophic, and they're also heterotrophic. So you have some that can make their own food, and some that have to eat from something else. How they move, there's a lot of variety in these 2 domains, and their life cycle, because they have only one cell, they are all asexual. Okay, now in eukarya, this is going to be chapters 18, 19, and 20, and then we're going to do a few more chapters in this class, but specifically these are how we're going to do the taxons. And so the cell type here, if you notice, they're all eukaryotic. Okay, how the other 2 domains are prokaryotic, these are all eukaryotic, and we'll go into definitions about that. The name eukarya is derived from their cell type because it's so cool about what they possess here. That's how their domain was named. And so under protista, they are unicellular with exceptions. Okay, so they all exist as one cell except for a few. Some of the types of nutrition, some of them are water and slime molds, and so these are heterotrophic, and they absorb their food. Algae, for example, that is not a plant, it is a protist, that is autotrophic, so it has photosynthesis that it undergoes to create its food. And then protozoa, these are the animal-like ones, and they ingest, kind of like what we do. How they move, there's a lot of variety, and their life cycle, they have a lot of variety. So this group, protists, I like to think of them as kind of bits of all the other animals and plants and fungi on one group that didn't quite belong. They weren't quite as evolved as the other ones, and so we threw them in this kingdom. Fungi is the same kind of deal here. They're all eukaryotic, multicellular with a few exceptions. So most of these are multicellular, and they are also heterotrophic by absorption. Okay, these guys do not move at all. They're non-modal, and they have sexual life cycle that is haploid, and we'll get into what that means later. Plantae are multicellular, all of them are. All of them undergo photosynthesis and are autotrophic. These also do not move, and they have sexual reproduction known as alteration of generations. Now, Animalia, they are all multicellular. They all are heterotrophic by ingestion, and we all undergo sexual reproduction, and it's diploid. And so we'll get into all those fun details in this class. Let's talk a little bit about the language of science. Mostly it's Latin, and there's some Greek thrown in as well. The reason for this is because Latin is a dead language. It's never going to be changing. It's always going to stay the same. And so throughout the course, I've decided to put important Latin-based roots in red, so hopefully that will help you guys know some of the important meanings of these words. In Latin, for example, pro means before. Eu means true. So when we talk about prokaryotes, for example, pro means before, and karyo, or keri, means nut or kernel in Greek. And so prokaryotic literally means before nut. And this is dealing with how this organism has evolved. It does not have a nucleus. And scientists at the time decided to call a nucleus a nut, so karyo, and so that's how it got its name. So prokaryotic cells do not have membrane-bound nucleus, and they lack organelles. They do have ribosomes, which is a special type of organelle, but it's not membrane-bound. Eukaryotic cells, U for true karyote, have a true nucleus and contain various organelles, including ribosomes. And so if you look at the pictures over here, this is a basic what a prokaryotic cell would look like, and down here is what a eukaryotic cell will look like. So in Latin, let's talk about these base roots. Uni means one, multi means many, autos means self or spontaneous, hetero means different, trophic means food. So if something is unicellular, it means it's one cell. Multicellular, many cells. Autotrophic means they make their own food. So autotrophic organisms create their own food source, usually by photosynthesis, which is with the sun, or chemiosynthesis, which is by chemicals. The other way that animals or plants or whatever is organized is heterotrophic, and this relies on the environment for food, so different food. There's different ways that these organisms take these food sources. Heterotrophics are either absorbers, which secrete out enzymes to break down food, and then they absorb it back in. These are also called saprotrophs or decomposers. Ingesters actually ingest their food, so what we do. Now let's talk about sexual versus asexual reproduction. Sexual reproduction involves a process of what we call meiosis and fertilization. A lot of this is learned in biology 114. So this takes two parents to produce one offspring. These offspring are not genetically identical to parents. They are different. Now with asexual, let's talk about the Latin root here. Whenever you have a standalone word and you use the letter A in front of it, it means without. So asexual means without sex. So in this process, this involves binary fission or mitosis. And this is only requires one parent needed to produce offspring. And these offspring are genetically identical. They are the exact copies. So this is kind of like cloning, if you will. And so you have two different types, sexual reproduction and asexual reproduction. Let's talk now about domain bacteria and domain archaea. These are very similar structurally. They're prokaryotic, they're unicellular. Their cell composition actually is what makes them different. That cell wall. Also, their plasma membranes have different lipid composition or fat composition. They also have RNA bases that are different. So, with all these differences, even though they're structurally the same, archaea is actually more related to domain eukarya than bacteria. So let's look at this chart over here, this diagram. Excuse me. So we all have this a way long time ago, common ancestor. And then you see there was a branch off that formed domain bacteria. And then the other branch was archaea. Now eukarya actually branches off from archaea. So that means that all these differences here are more related to eukarya than bacteria. So we are more related to archaea than bacteria. Most, if not all, archaea live in extreme environments. They are called extremophiles. Phile meaning love. And so we'll talk about those in a couple of chapters ahead. But thermophiles, acidophiles, halophiles, and methanogens are some of the most common archaea out there. Systematics. Systematics is a broad way to group organisms based on the evolutionary relatedness. Group organisms are grouped together based on organisms and how they're related to each other. I want to make this clear. Evolution. Evolution is a change in a population's genes over time. That's it. And this is what makes life forms more suited for their environment. So evolution changed in a population's genes over time. It is not on the individual. So one little individual cannot evolve. Has to be the complete entire population to make a difference. So phylogeny, another term, is evolutionary history of a group of organisms. Phylogenetic tree is a diagram that indicates common ancestors and lines of descent, or lineages. It's very similar to a cladogram. Common ancestor. Common ancestors are the ones that we look at that give rise to the new groups. Ancestral character, these are traits that is present in common ancestor and all the members that come after. So you have an ancestor had something, all the descendants have that trait afterward. A derived character is a trait that has evolved and is not present in the common ancestor, so it's a new trait, something that has evolved in the population. So looking at this phylogenetic tree, we can see that there's a common ancestor down here. And these are the descendants, 1 through 4. Now you have this first derived trait. So that means the ancestor does not have whatever this new trait is, but groups or descendants 2, 3, and 4 do. So the common ancestor and descendant 1 does not possess this specific purple trait. Now we have our second derived character, and this means that everybody from here up will have that trait, but the ancestors before do not have that. So only yellow trait gene is going to be found in groups 3 and 4. And if there was another evolutionary event, the third derived character, only those that come after this trait that occurred have this. And so 4 will have the purple, the yellow, and the red trait, whatever that is, but its ancestors do not possess them. Let's look at the different types of data collected to determine evolutionary history of organisms. The first thing that should come in your mind are records, the fossil records. These are evidence of the past. Now, there's some pros and cons. Pros, if a record is complete, we can trace that lineage through time. We can date it. However, most fossils are not a complete record due to environmental conditions. If you're a soft body, you're not going to fossilize well. And we can't see all the ancestral or derived traits just by looking at this fossil. However, if we do have a fossil, this is how you can kind of date it without doing all the chemical testing and radon and whatnot. But if you're looking at, let's say this is layers of dirt. The oldest is going to be the furthest down, the deepest, the one that's going to be covered over time by other sediments. So the older the fossil is, the deeper it's going to be. Let's talk about comparative anatomy. First, homo means same, ana means independent of, logos or logos means same thought or word, and morph means a form or structure. So we look at morphological data. It's essentially we're comparing anatomies, how the structure and the form compare to each other. So let's look at homologous structures. Homologous structures have similar anatomical structures due to common ancestry. So example would be vertebrate forelimbs. They are anatomically similar. They have the same bone structure, they're about the same placements, they look the same, and they function the same. Analogous structures have similar functions, but different in structure. However, they are not related by a common ancestor. So if you're looking at birds' wings or butterfly wings, they both fly. However, they don't have the same bone structure. Butterflies don't even have bones. And so there's a lot of differences in their actual anatomy. Another example would be a cactus and a spurge. They both have spines or quills, but they're very, very different in how they are formed and what actually is on the inside. So what I want you guys to take home is evolution can result in structural similarities in organisms that do not have a common ancestor. So it's important to avoid assumptions that similar structures are due to common ancestry. Let's look right here of evidence of comparative anatomy. These are homologous structures. If you look at these different animals, the box turtle, the dolphin, human, horse, the chicken, and the fruit bat, and you see right here is the list of the bones. Now they're almost in the same positions in all these animals. They're used as movement, getting around, carrying something. They all have very, very similar placement, and they have similar what their jobs are, movement. And so that is why we look at to see the comparative anatomy. And these are homologous. They have very same, similar traits, which is indicative of a common ancestor. Now let's look at this one of analogous traits. Here's a moth, pterodactyl, and bird, and a bat. Looking at these three in the bottom here, you can see the bones. They're similar. And as you can tell, dinosaurs look a little bit different than bats because things have evolved. However, they still have a lot of the same bones, and they all are used to fly. Now looking at this moth here, it also flies, but there's no bone structure. And so it's safe to say that somewhere on the line, these ancestors diverged a long, long time ago. And so the bat and the bird and the pterodactyl are more related because of their comparative anatomy is homologous versus the moth, which is analogous. They both fly, but use very different methods of actually how their body works. Okay, so looking at now molecular studies, we have DNA and RNA-based sequencing. We have something called what's a molecular clock. Now this is change over time at the DNA level, and it's predictable. Okay, so it's like evolution, but we're checking just at that DNA and how it changes over time. So if you go into more studies with this, you'll learn more about it. But lineages with similar genes are more closely related and have a common ancestor. So if you're a bird and a bat, your genes are going to be more related and more similar than, let's say, a moth. I mean, they're all animals, but the bird and the bat look more the same, and their genetic level, now looking at the molecular data, indicates that they are more similar. So taxonomy is ever-changing due to technologies that are being advanced, and especially in the molecular field. So this is a very cool field because it's always going to be changing, always going to be updating. We find new things, we develop things, and it's just going to always be changing. So maybe in a couple of years from now, we're going to have different research to teach you guys. All right, so let's talk about cladistics. Now, cladistics is a method of determining evolutionary relationships when a complete fossil record is not available, which is most of the time. So we use what's called a cladogram. It's kind of similar to a phylogenetic tree, and it depicts the evolutionary history of a group based on the data that we have. Okay, but we have less information than when we use the phylogeny. So we have something called outgroups and ingroups. So outgroups are species that defines which study group trait is the oldest. And ingroups is our species that will be placed into clades, or groups, right, in the cladogram. So coming down here and looking at this cladogram, we have the outgroup and the ingroup. So everyone that we're looking with the ingroup has those same traits. The ones in the outgroup do not, and so that's how this is working here. So ancestral trait. Now these are traits present in both the outgroup and the ingroup. So these are old, everybody from here on up has that trait. The clade is a branch, okay, of a cladogram that contains common ancestor and all of its descendants, okay? So like the branch. Shared derived traits, these are traits that are distinguished at a particular clade. Now parsimony, this results in the simplest cladogram possible. Now if you think about parsimony in evolution, you want to make sure that the easiest way for something to evolve has occurred. Because if you have a lot of evolution going on, it's not very likely. And so having one chance of a change happening to affect the rest is more likely than having multiple chances. So looking at this picture down here with these birds, so red feathers. Having red feathers evolve twice isn't as likely as having it evolve once. And so this is more preferred because you have this trait here saying that, okay, instead of having two individual events of evolution occurring, we had one that affected every group after, all the descendants. So that's what parsimony means. All right, so we're going to take a fun look here at the lamprey, shark, and salamander. We're talking about the lizard, tiger, and gorilla. Okay, so we're going to make our own cladogram here. And we're looking at specific traits: jaws, lungs, amniotic membranes, hair, if there's a tail or not, and if they're bipedal. Now, the clades, so we're talking about how these share a common ancestor, and we're looking how much they relate to each other by these traits they have. And so, lampreys don't have jaws, okay, but everybody else does. Then lungs. Sharks don't have lungs, okay, neither do lampreys, but everybody else does after that. The amniotic membrane, this is dealing with eggs that that keeps eggs moist, and so reptiles have evolved. They don't have to be next to water to lay eggs. And so from reptiles on down, they have this amniotic membrane. Now, tigers are the first ones in our list to have hair, and gorillas don't have tails, neither do humans, and we're the only ones that are bipedal. So if you look at this, you can say, all right, everybody has, except for lamprey, has jaws. And you see it gets more and more specific as you go down, and I had this chart all nice, but if you were going to do it, you probably wouldn't know exactly where to put these traits, in what order. And so you might have had bipedal on one side or the other if you were, this is generally looking at the DNA, so you wouldn't know exactly what they were. But this is just a fun way to really see kind of what work is done. And so I want you guys to think, what could be an ancestral trait that is shared among all these organisms? Look at the picture real quick. I would say an easy one would be eyes of some kind. That's one to think about. Okay, so now that we know we have this chart filled out, let's actually make a cladogram. And so we know that lamprey, of all the ones listed of our traits, it didn't have anything. So that has to be one of the older ones because it hadn't evolved any of them. So you put that down there. Now, with jaws, that was what everybody else had after. So that's the trait that evolved after lampreys were created. And so the same thing with shark. Okay, lungs. So from here up, everybody else has lungs. The salamander didn't have the amniotic membrane, but everybody else did, and so forth. So that's how you actually create a cladogram. So we're using it can be use so much different kinds of data. You have physical structures, DNA sequencing, you can even do behavior, and there's a lot of different things you can do to construct a cladogram depending on what you're looking at. So here's the same chart, just visually looking at it, so you can kind of see how they are. Okay, so evolutionary systematics versus phylogenetic cladistics. So you have two different viewpoints used to group organisms based on their evolutionary relatedness. Now, it's the same data, but it's interpreted differently. So evolutionary systematics, we use the Linnaean system, and it stresses both the common ancestry and the degree of structural differences of these divergent groups. Phylogenetic cladistics generates those clades, or those groups, based on only using shared traits. So this is kind of the newer philosophy because we don't have all the information from history. And so we're using what we have to construct relatedness. So the problems with these two. It's difficult to compare clades with taxons, and taxons are not necessarily equivalent in the Linnaean system. You have a phylum taxon in plants versus animals, it could be different. And so we're kind of going more towards the phylogenetic cladistics area, but from years and years we've used evolutionary systematics. So we're trying right now to see how we can use both of the information to the best that we have. And hopefully this has been helpful, and let me know if you guys need anything else.